So good afternoon and welcome. Thank you for taking the time to join us for this special research update. Gathered for the live presentation today are members of the Hearing Health Foundation Board of Directors, staff, and some of the Hearing Restoration Project Consortium scientists who were kind enough to step away from their microscopes to join us today. We have people with a range of knowledge of hearing loss research listening in. For some of you, this is an introduction. Others have heard a little bit about HRP from a colleague, friend, or loved one, and are participating today to learn more. Some of you have a deeper level of understanding of our research and are looking for an update on hair cell regeneration, the HRP progress made to date, and where we're heading in the months ahead. No matter who you are, we all have one thing in common, a personal connection to hearing loss. Maybe you or a loved one was born with a genetic hearing loss or developed hearing loss over time due to loud noise or even instantly from a traumatic event. And some of you have hearing loss or know someone who does because it happens to us as we get older. We want everyone to obtain new knowledge and information today, so Dr. Bar Gillespie will cover a lot of ground over the next 30 to 40 minutes. Some of the scientific terms, visuals, and materials presented to you may be unclear. Dr. Bar Gillespie will provide definitions as he speaks, but please write down whatever questions you might have. And just assume that other participants want to know the same thing and will be grateful to hear the answer. There will be time for Q&A at the end of the presentation, so please don't be shy about asking questions. Our goal is that everyone who listens to this presentation will obtain new information on hearing loss and our research toward a cure. And it doesn't end today. If, after listening to the briefing, time passes and you have questions or thoughts, we are your source for reliable, accurate, up-to-date information. We intend to hold many more of these special research updates with our researchers in the future. There will be plenty of opportunities for learning as we go forward. Thank you again for your time and interest in our Hearing Restoration Project. I want to just take a moment or two to give a brief bit of history of Hearing Health Foundation in case you're unaware. Hearing Health Foundation was founded in 1958, almost 60 years ago, and we've established a reputation for pioneering breakthroughs in hearing and balance research. Some examples of this were we were early supporters of the revolutionary cochlear implant, and today, over 220,000 children and now adults are benefiting. We advocated for the passage of the universal newborn hearing screening legislation in the 90s, and today 97% of newborns are tested for hearing loss at birth. Throughout our history, our Emerging Research Grants program has and continues to provide seed funding for early-stage scientists in hearing and balance science, leading to discoveries in hair cell regeneration, tinnitus, hyperacusis, and Meniere's, to name a few. Many of these researchers have had fruitful careers and gone on to obtain federal funding for their work, and this is one of the measures of success. Our experience throughout the years leads us to our focus today, a biologic cure for hearing loss and tinnitus through hair cell regeneration, our Hearing Restoration Project. And now I'd like to turn it over to Dr. Bar Gillespie, our HRP Director and Professor at Oregon Health and Science University. Peter, take it away.
Claire, hello and good afternoon to everybody. I'm going to mute everybody right now.
The conference is in lecture mode.
So I want to tell you today about the Hearing Restoration Project, both the motivation behind it, the approaches that we're taking, and the progress we've made to date. And I hope that I'll give you some new information that gets you excited about where we're going with this program. First, though, I want to give you a little bit of background on the ear, just so that we're all on the same page. I think most of you know about the external ear here, and probably most of you know that the cells that are actually responsive to sound, the so-called hair cells, live in the internal ear here. And in fact, the internal ear is made up of two divisions. the vestibular system, which I might talk about in a moment, and that's used to give your sense of position of your head in space, but also the auditory system, which is responsible for detecting sound. Now, I'd like to just give you a sense of where these hair cells are, but don't worry about remembering all the details. It took me a couple of years to really understand the anatomy of the organ, but I think it's still useful to go through. Here's a cruder view of the ear on the left, and if you take a cross section through the part of the inner ear that's responsible for detecting sound, the cochlea, you see a complex system here of multiple chambers. But the key thing are these red guys here, the so-called hair cells. They're the sensory cells that respond to sound. And they're also the cells that are uniquely vulnerable to damage. There are other cells around the hair cells that I'll say more about later. The hair cells have a very specialized component at their apical, or their top of their cell, called the hair bundle, and that's the kind of antenna that responds to sound. And I won't talk about the details of it, but just suffice it to say that we think that in noise damage, that this hair bundle can be damaged and the hair cell then dies afterwards. So here's a picture of a real hair cell. You're actually just looking at the top of the hair cell with all of the hair bundle on top. So the green part here is one hair cell, and you can see the excuse me, part of the cell that responds. The cell itself goes down deep below the plane of the picture here. You can also see that the cell is surrounded by other biological material, and that's the so-called supporting cells, which I'll talk about later. These are really crucial because we think supporting cells may have the capability of turning into hair cells eventually, and we know that's true in some species. So I've already told you that hair cells are destroyed by noise, and this just gives an example. These are from a cochlea. These white things here, these V-shaped structures, are the hair bundles of the cochlear hair cells. And you can see after noise that many of them are missing completely, and the hair cells actually have died. Likewise, other treatments, other conditions that lead to loss of hearing, like some types of antibiotics, some types of cancer chemotherapeutic agents, and even just aging leads similarly to the loss of the hair cells. So losing hair cells means you lose your hearing, and we'd like to get them back. So in the past century, the primary treatment for hearing loss has been hearing aids and cochlear implants. And these have been very successful, but certainly are imperfect. I think for this century that we've got a number of different thrusts into getting much more effective therapy. First of all, and foremost, I would say, is prevention of hair cell loss. Preventing different investigators, plus myself, the scientific director. And one of the key things, and I'm going to come back to this a couple of times, one of the key things about the scientific advisory board consisting of a number of different scientists from across the country. And the scientific advisory board gives us oversight, gives the HRP, sort of a check. They evaluate our proposals and they give us guidance as to directions in which to go. So this slide I'm not going to belabor, but I just want to point out that there are 15 of us in the HRP from across the United States, and in fact international. There's one member from Canada and one member from Great Britain, and so we've brought together the best of the auditory field in order to bring our minds together and be able to do things that we can't do on our own. So one of the key facets of the Hearing Restoration Project's approach is that we use 3 different animal models for studying hair cell regeneration. And we do that for a couple of interesting reasons. First of all, 2 of those models, the chick, as you see here on the left, and the zebrafish, as you see here on the right, they show robust hair cell regeneration. So if you damage the hair cells of a chick or of a fish, then within a short time, only a day or 2 for the fish and a few days to a few weeks for the chick, the hair cells are come back. New hair cells are formed. So that's really spectacular because it tells us that animals are capable of regenerating hair cells. By contrast, the mouse is our other experimental model, and it serves a similar role. It stands in for people. So the mouse shows no hair cell regeneration after a few days after birth. You can damage the hair cells in the mouse, and as far as we can tell, nothing much happens in terms of restoring hair cells. And so we can figure out how to regenerate hair cells in the mouse. Then I think we can figure out how to regenerate hair cells in people. So let's look a little more in detail at the inner ear, because there's a couple of features here that will give you an idea of how we're trying to go about this project. So I mentioned supporting cells. So here you can see both hair cells in red and supporting cells in yellow. And the supporting cells surround the hair cells. They play a structural role, as their name would seem to indicate. And you can see they're not homogeneous. There are different types of hair cells that do different things in the ear. So we know from experiments that have been done in the past that what happens when fish and chick have damage to their hair cells is that it's the supporting cells that give rise to new hair cells. Either a supporting cell can turn directly into a hair cell, or it can divide and make one hair cell, and the other cell would remain a supporting cell. However, in the mouse, that doesn't seem to be the case. Supporting cells don't convert to hair cells after the hair cells are damaged. So we're interested in understanding what the molecular signatures are of the supporting cells. How do supporting cells that can turn into hair cells, how do they differ from those that cannot turn into hair cells? And what are the molecular differences? Because those are things that we potentially could tweak. We also want to know what happens to the supporting cells in the mouse after damage to the cochlea. If the whole cochlea degenerated and we were left with nothing, then it would be much more difficult to stimulate regeneration of hair cells. In the cells to make new hair cells. And when I say pathways, I mean sequences of molecular reactions that take place in the cell. There actually are hundreds or even thousands of pathways that are operative simultaneously in a cell. We think that there are likely to be a few of those pathways that are important for hair cell regeneration. Maybe in the mouse, there are pathways that inhibit regeneration, and we need to interfere with those pathways. Or maybe there's a pathway that's active in chick and fish, and to see if we can track down whether there are pathways that are stimulatory or inhibitory towards hair cell regeneration. And then we also want to figure out whether or not the supporting cells remain behind or we have that flat epithelium. The second phase is crucial because the experiments that come out of the first phase are not So one of the most important sets of experiments that we've done in the past 3 years is so-called genomic profiling in these 3 model systems in response to hair cell damage. We want to ask whether or not genes are turned on or turned off in response to that damage to hair cells. And in particular, we want to understand why the mouse doesn't show the same type of responses as the fish and the chick. So we've made a lot of progress with this step. We've generated large data sets from each species. We've done multiple types of experiments with each species. And then we've used a strategy, a technique called bioinformatics, which is using computational methods that are coupled with biological information to try and understand the relevance of the data that we've generated. Just having large lists of genes is not terribly informative. We need to understand which of the genes are play together in a pathway, and which of the genes might be relevant for regeneration. And so these bioinformatics experiments are crucial for that. The other part of phase 1, and this should be a B right here clearly, is to understand what the B should be right here. The second part is to understand the supporting cell fate. Do we get supporting cells remaining or a flat epithelium? And I would say that our experiments look pretty good, and they suggest pretty strongly that at least in mouse, even after many months after damage to the hair cells, the highly specialized supporting cells remain. And that is nice. It corroborates some data that have been gleaned from studying human ears, which suggest the same. So this is important because it suggests that we can target these supporting cells, which on a molecular level are much more closely related to the hair cells than these flat epithelium cells. Phase 2, as I said, we're going to be modulating the genes of interest and asking whether we can turn on regeneration in the mouse or maybe turn off regeneration in the fish and the chick. And we've got a variety of new projects that are starting on phase 2. And I'm going to briefly talk about the projects that are funded for this year in a couple of slides. So we haven't started phase 3 yet. We really need to get a healthy way through phase 2 for 2 reasons. One, we need to make sure that we have the ideal model system for carrying out the experiments, which are going to be time-consuming, expensive, and technically challenging. And then also, we need to be sure that we know what we're targeting, because the phase 3 experiments could be much more directed if we're pretty sure we know which pathways are important. So I just want to reiterate the point about collaboration. So a lot of what we've done so far to date really has depended on the ability to speak together. We have an annual meeting for a couple of days, and we have another meeting associated with the meeting that all the scientists go to. Moreover, we talk on the phone at least monthly and oftentimes much more than that. And this allows us to be continuously collaborating together, sharing ideas, and it's really delightful from a scientist's point of view and tremendously productive. So there are simple things we can do with collaboration. We can not all do the same experiment because we're not talking to each other. Instead, we can assign, give an experiment to a different person. The collaboration also allows us to do these multi-species, multi-model experiments, and that really couldn't be done in any one lab. And that's really crucial to our approach. I think it's been very successful so far. And then finally, the collaboration allows us to see the new data months or even years before you would if we had to wait for it to be published. And the collaboration is working. One measure is just to look at who comes together to propose projects to be funded by the HRP within our group. And almost all of the projects involve multiple scientists, usually from different institutions, coming together to propose an experimental approach. And I think it's a good test, a good example of how this is working. So I think we have made substantial progress on this first phase. We've identified a variety of candidates for these molecular triggers of hair cell regeneration and the pathways that are necessary. Well, we have too many, so we really are continuing to use bioinformatics methods to glean through and determine which are most important. We have definitively shown, I think, that at least in the mouse, the specialized supporting cells remain. So we know now what our target cells are for triggering hair. That multiple people in our consortium had seen the same phenomenon, which they each had said, that's interesting, but I'm not sure if I believe it. And the phenomenon was they damaged hair cells in the mouse. They have responded to the damage to hair cells in the mouse. They've gone partway along the pathway towards making a new hair cell, but they're stuck somewhere. And we'd like to know where they're stuck, because that would be those species that show it or how it doesn't work in the mouse. We have clues that are pointing us in the right direction towards overcoming it, but we have a lot more to learn. So these other projects, such as a limited amount of funds to do these phase 2 projects. They're going to take much more than we have right now. But we've gotten started both on actually screening some of the molecules and pathways, but also in developing tools and techniques that allow us to do this better in the future. So Dave Rabel and Andy Groves and Jenny Stone have a project that couples together fish and chicken and mouse and both candidate molecules and pathways, but also drugs to look at hair cell regeneration. Josh Raphael is developing new ways of delivering genes in the mouse. What we call low throughput, which means they're laborious, one molecule at a time. It's going to take us a while to get through large numbers of molecules and pathways. But it's necessary to do it this way because it's really what we have right now. But our phase 2 must be scaled up, and this is going to require quite a bit what we've learned over really the past 25 years or so is that hair cell regeneration is indeed a plausible way in which to eventually treat hearing and balance disorders. It's not going to give us hearing restoration next year, but it will. Now to answer any questions that anybody has, either sent in on the Adobe Connect or spoken over the phone. Thank you.
Hello, this is Brian.
Synapses. And so there's a recognition that a substantial fraction of hearing loss may be due not just to damage to hair cells per se, but maybe just to damage to the synapses. So our cells.
All right, thank you.
Any other And I think that the universities of the scientists are going to be interested in having a role. But I also think that one of the things that I have not mentioned is that we anticipate, as we get closer to having actual therapies or having drugs that we can look at, that we're going to need to be partnering with industry. And because they have the resources, the big resources, once a lead compound has been identified, they have the
research toward a cure, our progress to date, and where we're going as we move forward. So what I would say is after listening, and while time passes, if you have questions, thoughts, things come to mind, don't be afraid to contact us. We are the source for reliable, accurate, up-to-date information, in particular in the area of hair cell regeneration.